So tuberculosis is the disease we'll talk about next. So this is a communicable disease of the lungs and respiratory tracts, and this is caused by Mycobacterium tuberculosis. Tuberculosis is spread from person to person by the airborne route. So in children, the lung is the first site of infection, and most infections resolve with some local scarring. So this is what's called the primary complex. Then in about 5%, the infection is not controlled and it spreads from the primary focus through the body, which we call the miliary spread. This may resolve, or this could develop into other infections such as meningitis. Disease may also emerge if immunity wanes, for example, later in life. Immunity depends on effective T cell function, so if compromised, individuals are more likely to develop symptomatic disease. So symptoms of tuberculosis include persistent cough, chest pain, loss of weight. But the good thing is, with treatment, TB can almost always be cured, but they need quite a long duration of treatment. So combination therapy, usually 2 antibiotics for 6 months. We also have the BCG vaccine available in the UK, but this is used for groups that are considered at risk of getting tuberculosis. It isn't used in over 35s because there's no evidence that it works in that group of people. So the next disease we'll talk about is cholera. So this is a highly communicable, which also means contagious disease. It's caused by the gram-negative bacteria Vibrio cholerae. And the bacteria is transmitted by contaminated food and water, usually found in developing countries where there's inadequate sanitation and an unsafe water supply. So when the bacteria are ingested through contaminated water or food, bacteria are able to survive the acidity of the stomach. And they're able to attach to the intestinal epithelial cells. They then produce the cholera toxin, which results in secretion of water and electrolytes into the lumen of the bowel, so into the space of the bowel, causing diarrhea. So the main symptoms of cholera are massive painless fluid diarrhea, so up to 20 litres a day, which is massive, and vomiting. And this, as you can imagine, results in severe dehydration and electrolyte imbalance. It is a highly serious disease which can lead to death within 24 hours. So treatment is with oral rehydration solution, so salt and glucose mix. If it's really bad, obviously they might not be able to keep down the oral rehydration fluid, might not be sufficient, so then they might need IV fluids, so intravenous fluids. Antibiotics such as tetracycline or ciprofloxacin can also decrease the duration and severity of the disease. So in this country, you can get cholera vaccination if you are deemed at high risk. So if you're traveling into a country where they've got unsafe water or not very sanitary environment. And the next disease we'll look at is typhoid, also called typhoid fever. And this is caused by a bacterium called Salmonella typhi. And this is related to the bacteria that cause salmonella food poisoning. So this is also transmitted through contaminated food. It's highly contagious, and the infected person can pass the bacteria out of their body into their stools, their poo, or less commonly in their urine. And if someone else eats food or drinks water that's been contaminated with a small amount of the infected urine or poo, they can then become infected with the bacteria and develop typhoid fever. This could occur, for example, if an infected person has not washed their hands after going to the toilet, and then they prepare food for someone else, as an example. So when a person ingests the Salmonella typhi bacteria, it invades the intestinal wall and spreads to local lymph nodes before reaching the blood. And once the bacteria in the blood, in large numbers, this can cause massive problems because they can then potentially affect many different organs in the body. So the symptoms of typhoid include fever, headache, abdominal tenderness, constipation, red spots in the body, and hemorrhage in the intestine. But typhoid is usually treated successfully if treatment is prompt with antibiotics, for example, ciprofloxacin. Typhoid can be prevented by avoiding contaminated foods, by destroying bacteria by heat and refrigeration. However, typhoid is uncommon in the UK and occurs mostly in developing countries where sanitation is poor and they don't have clean water. Children are thought to be most at risk of typhoid just because their immune system is still developing. There is a vaccine available to prevent typhoid that you can get here, and you might need this if you're traveling somewhere where sanitation, food hygiene is likely to be poor. Okay, so the next disease is gonorrhea, and this is caused by the gram-negative Diplococcus neisseria gonorrhea. And this is most common in individuals between 15 and 35 years of age. And it's an STI, so a sexually transmitted infection, and it's almost exclusively spread by sexual contact. So the way the bacteria cause the infection is they adhere to the genitourinary epithelium via pili. If you remember pili from the first lecture, so these are the hair-like appendage on the surface of the bacteria. So these pili help the bacteria to attach to the epithelium of the genitourinary tract, and then they can invade the epithelium layer and produce an inflammatory response. So the symptoms in women are an unusual discharge, maybe greenish or yellow in colour, pain or burning sensation when passing urine, pain or tenderness in the lower abdomen area, and bleeding between periods. In men, they can also get unusual discharge on the penis and a burning pain when urinating. They could also get inflammation of the foreskin or pain or tenderness in the testicles, although that testicular pain is rare. So both men and women can develop an infection in the rectum, throat, or eyes by carrying out unprotected anal or oral sex. If infected semen or vaginal fluid comes into contact with the eyes, you can also develop conjunctivitis. So gonorrhea can be treated by antibiotics, so usually either intramuscular ceftriaxone or oral ciprofloxacin are used to treat gonorrhea. But however, the exact treatment will depend on whether the antimicrobial susceptibility is known. So you remember we talked about antimicrobial susceptibility testing in the last lecture. So this is of increasing importance with gonorrhea because gonorrhea is starting to become resistant to various antibiotics. So it's important to know if you're prescribed an antibiotic that it's actually going to be working against bacteria. So prevention of gonorrhea can be prevented by avoiding sexual contact with individuals at high risk and using effective barrier contraception. It's also important with gonorrhea that if you've been tested and diagnosed, that contacts have infected individuals are traced and treated as well. Okay, so the sexual partners are also treated. So next up is botulism. So botulism is a serious paralytic illness caused by a nerve toxin, which is produced by the bacteria Clostridium botulinum. So botulism is mainly due to ingestion of contaminated food, although human botulism can also be caused by intestinal infection with C. botulinum in infants, by wound infections, and by inhalation. But the main route is through food. So foodborne botulism occurs due to incomplete heat treatment of contaminated food during the canning or bottling process. So because it's not being killed by the heat treatment, bacteria is able to survive and produce the toxin botulinum toxin. So this toxin is a very potent neurotoxin. Okay, it's one of the most powerful toxins known in science. And when it's ingested, it affects the nerve endings and neurotransmission, resulting in muscle paralysis. This results in paralysis of the eyelids, also difficulties in speech, breathing, and swallowing due to this muscle paralysis. And treatment is with a specific antitoxin to neutralize the toxin and breathing support. And this condition can be fatal in certain cases. As a result of the high food standards in the UK, the chances of getting foodborne botulism are extremely small, but following food hygiene procedures will reduce any risk. So it's advised that you don't eat any food from bulging or damaged cans or tins, and avoid eating foul-smelling preserved foods, food stored at the incorrect temperature, and out-of-date foods. Heroin users are also at risk of wound botulism through injecting contaminated drugs, so they should avoid injecting into the muscle. In addition, you should also avoid giving babies of under 1 honey, as this is known to contain Clostridium botulinum spores. Okay, so now we're going to touch a bit on antibiotic treatment. Okay, not too much detail, just a bit of an introduction. So antibiotics are specific to bacteria, so they specifically treat bacteria, so they don't treat viruses. So you might have heard people say, oh, don't take antibiotics for a cold, because the cold is a virus. So yeah, these are specific for bacteria. And antibiotics themselves are products of fungi or bacteria. They are categorized as having a broad, narrow, or medium spectrum of action. So you might have heard the phrase broad spectrum antibiotics. So these are your penicillins. These are antibiotics which work on a broad range of bacteria, so a host of different bacterial diseases. And then you've got narrow spectrum antibiotics which just work on a few species of bacteria. And in terms of antibiotic efficacy, we can categorize them as bactericidal, so they actually kill the bacterial cells, or bacteriostatic, where they inhibit the growth of bacteria. And they have many different mechanisms of action, and we're going to just look at a few of them today. Okay, so now we're going to look at the different types of antibiotic treatment available. So they can be categorized into different groups. So first of all, you have the beta-lactams, which are the penicillins and cephalosporins, and these are very commonly prescribed. And these inhibit the cross-linking of the peptidoglycan in bacterial cell walls. So in this way, they inhibit bacterial cell wall synthesis. Then you have rifamycin, and this works by inhibiting the DNA-dependent RNA polymerase. Okay, and by inhibiting this enzyme, prevents transcription of DNA to RNA. So bacteria basically can't transcribe any of their genes to be able to carry out their normal functions. Then you've got aminoglycosides, tetracyclines, and macrolides, and these block translation and therefore protein synthesis in bacteria cells. Quinolones, on the other hand, inhibit bacterial topoisomerases, which interferes with DNA replication and transcription. And the last ones we're mentioning here are the sulfonamides and trimethoprim, and these inhibit the synthesis of tetrahydrofolate. So tetrahydrofolate is very important for bacteria as it's the precursor of folinic acid and ultimately purine and DNA synthesis. Now we're just going to discuss a few of the important resistant mechanisms. So, as we mentioned bacterial resistance before, we'll just mention it now because it is very important due to the overuse of antibiotics, bacteria becoming more resistant. So we define resistance as when a previously susceptible bacteria is no longer inhibited by an antibiotic at concentrations that can be achieved safely in the clinic. Okay, so it's when a bacteria was sensitive to the antibiotic, the antibiotic was working on it, and then the bacteria acquired resistance. The antibiotic no longer works. So some of the important resistant mechanisms are limiting uptake of a drug. So some bacteria create barriers to stop the antibiotics entering the cells. So that's one mechanism. Another mechanism they can acquire is modification of a drug target. So as we've mentioned, a lot of these antibiotics target enzymes, bacterial enzymes. So bacteria can acquire mutations, basically modify the structure of the enzyme slightly. So the natural substrate can still bind, so the enzyme can carry out its normal activity, but the antibiotics can't bind anymore because it's slightly changed. Okay, so then the antibiotics are no longer effective against those bacteria. Another mechanism is inactivation of a drug. So this is a common resistance mechanism. So for example, many strains of Staphylococcus aureus produce an enzyme called beta-lactamase, and this can break open the beta-lactam ring of penicillin and therefore inactivate it. And bacteria can also develop resistance by acquiring a protein that actively pumps out antibiotics. We call this an efflux pump. So now we're going to look at antibiotic susceptibility testing or antimicrobial susceptibility testing. And this is the DISC test that we talked about in the earlier lecture. So I'm just going to go into a bit more detail on this. So as we said, to do this test, you spread the bacteria on the agar plate, and then you place the paper discs impregnated with antibiotic onto the plate with the bacteria. And then after incubation, the growth of the bacteria is observed, and areas around the antibiotic disc where no bacterial growth can be seen are called zones of inhibition. And these zones show that an antibiotic has been successful in stopping bacterial growth or killing the bacteria. So the larger the zone of inhibition, the more sensitive the bacteria are to the antibiotic in the disc. And the size of the zone of inhibition is inversely related to the minimum inhibitory concentration, which is the lowest concentration of antibiotic required to prevent bacterial growth in an overnight culture. The minimum inhibitory concentration can be calculated from standard curve graphs based on the diameter of the observed inhibition zone. Okay, so this slide gives you a nice timeline of when the different groups of antibiotics were synthesized first. And it also tells you which antibiotics are bacteriostatic and which are bactericidal. And as we said, bacteriostatic means that they inhibit the growth and reproduction. And then bactericidal means they actually kill the bacteria. So as you can see, the yellowy-green antibiotic groups, such as chloramphenicol, sulfonamides, are commonly bacteriostatic, whilst others, such as the beta-lactams and aminoglycosides, are commonly bactericidal. Okay, so just to summarize, we've established that humans live in a microbial environment, so microbes are everywhere, they're on our body, in our body surfaces, so they're everywhere, we can't get away from them, and some of them are actually beneficial for us. And we've also looked at the normal microflora in the different areas of the body, so the GI tract, for example, the urinary tract, and we've also discussed how, although many of the bacteriophora are harmless, some can cause disease when conditions are favourable. And we've looked at a few examples of bacterial diseases, so some clinically significant ones. We've looked at how the disease occurs, okay, the pathogenesis, and we've looked at the clinical features and symptoms of the disease and how it's managed. We've also touched on antibiotics as well, some antibiotic mechanisms of action, and we've also looked at some mechanisms of resistance as well. And we've looked at specific terminology such as bactericidal, which means to kill bacterial cells, bacteriostatic, which means to inhibit growth. Okay, so here's a list of useful textbooks. So I would advise you do some further reading around the topics so you get a better grip on the subject, okay? So it'll help your understanding. So it's always good to do a bit of reading around the slides. Okay, so that's the end of the lecture, and I look forward to the next one.